All right. Good afternoon, everyone. Welcome everybody to this week's immuno-webinars. So it's a great pleasure for us to receive here Luísa Menezes Silva, who is a there was a delay in the sound here, so I think now it's fine. So it's a pleasure for us to receive Luísa Menezes Silva, who is a PhD student in our program, but she recently just published a very big paper. So it's going to be a pleasure for us to know, to listen better about this story that she's ready to tell us. Thank you very much, Luísa, and the floor is yours.
Thank you so much, Diego. And first of all, I would like to thank the organizers because a few years ago, I was helping to organize the immuno webinars with Gustavo and a little bit with Diego as well. So now I'm at the other side, right? So very happy for being here. And thank you all for coming to this presentation as well. So for those who don't know me, I am Luísa Menezes. I'm here from the Department of Immunology of the ICB-USP. And this work I'm going to present today, it's a result from my abroad fellowship at the Royal Cornell Medicine in collaboration here with the ICB-USP at the University of São Paulo. So basically, I did most of these results abroad in collaboration with the Jill Roberts Institute for Research in Inflammatory Bowel Disease in New York and the Friedman Center for Nutrition and Inflammation at the laboratory of Professor Nick Collins. And here in Brazil, I did this study at the laboratory of Professor Nils Câmara. So basically, what I'm going to show you today, it's the innate and adaptive immune rewiring in response to nutritional fluctuation. So first of all, I would like to introduce the concept of food fluctuation because we and our immune system that uses a lot of nutrients and energy, we have evolved in the context of fluctuating food and nutrient availability. Because we have to think that a lot of exogenous and endogenous factors, they can limit how the immune system and other cells within our body can use and find nutrients. So for example, there are a lot of events as drought regions or desert regions, some natural disasters, the change in the seasons, and even some infections that can be very energy demanding for the body and also impact the immune system. And other factors such as cancer as well, endogenous factors that can limit how the nutrients and calories they are available in the bloodstream. So the major curiosity we had at the beginning of this project was what mechanisms have evolved to support the immune response when calories and nutrients they are running low. So at the beginning of this project, we focused in one niche of the adaptive immunity, T cells, for two reasons. The first one, it's because they are important for fighting infections and controlling antigens from the beginning. And also because they generate memory after all. So just a reminder about how the T cell activation occurs. When you have a naive T cell and it recognizes its cognate antigen for the first time, which we call a primary challenge, it has a rapid expansion and become a lot of effector T cells producing a lot of cytokines and other factors. But then after controlling that antigen, there is a contraction of the immune response. And at the end of this period, there must remain some T cells that could generate memory cells. So then they stay, especially at the bone marrow at this time. And if in case of they find the same antigen in the future, they're gonna respond in which we call a secondary challenge, so that here there is a difference because now the cells, they are poised to function, so they act even better and they produce cytokines and other factors very fastly and more efficiently. So the Yasmin Belkite group and Nick Collins group, they have published recently that if you put some mice into a dietary restriction, which means that these mice, they are eating less than the control ad libitum mice, however, they are not malnourished, which is important to say. The CD8 T cells and CD4 T cells, they have better response against a bacterial infection. So their response is 500-fold better in the protection of this bacteria. And the cells, they are producing way more cytokines as well as interferon gamma. And this is mediated in part by the microbiota because they can activate better the disease in the gut and in the lymph nodes. However, let's think about the amount of energy that is necessary for activating these cells. Because in a secondary challenge, as I said before, once the cells they are already poised to the function, they don't require a lot of energy. They have maturated the affinity, so they are more mitochondrially based. They use way less energy when compared to the primary challenge. You have a lot of clones to the same antigen, you proliferate a lot. One of these quiescent naive T cells, they can expand up to 20,000 effector T cells. So this uses a lot of energy, especially glucose in this time point. And a conceptual problem we had at the beginning by using this data restricted mice in these experiments was knowing from the beginning that data restricted mice, they have lower blood glucose and lower nutrients, not only in the bloodstream, but also in the tissues. So the main question for this project was, how will the host navigate an energy demanding effector T cell response during a dietary restriction? This question was way more intriguing because, especially for the lymphocyte compartment, that was the focus at the beginning, because some of the first results we got, looking for this kinetics from from week 1 to week 6, at a dietary restriction, reducing the amount of nutrients these black 6 mice they were eating, was that from day 1 to week from week 1 to week 6, they have a substantial reduction of the CD8 and the CD4 compartments at the periphery, even in the blood, in the lymph nodes, or in the spleens. And in the other hand, the cells, they are increasing in the bone marrow, showing that somehow they are just going away from that nutrient, the lack of nutrients in the periphery, and going to a safer hub that in this case is in the bone marrow. But, well, the responses, they are mainly architected in the periphery. So I go back to the question from the beginning, how will the host navigate an energy demanding effector T cell response? So to answer this question, we put some black 6 mice onto 2 different dietary protocols, ad libitum, which means free access to food all the time, or dietary restriction, which means eating 50% less compared to the control group, ad libitum. And during 3 weeks, they went into these dietary protocols, and after that, we infected these mice with this foodborne pathogen, Yersinia pseudotuberculosis. We used this pathogen for 2 main reasons. The first of them, it's because it can spread to the periphery and then cause a systemic infection. And the second of them, it's because it activates a strong CD8 cell response. So it was important for the what we wanted to look from the beginning. And after 8 days, which means the peak of the active infection for this type of bacteria, we took the mice down and we looked the parameters. So something that got our attention is that in spite of the low glucose levels these mice have from the beginning at steady state, and which is critical for sustaining the immune responses, you can see there is no difference in the survival curve here for adult mice or dietary restricted mice, or even in the host protection showed by the bacterial burden in different tissues at these mice. And this shows that the protection against this pathogen is granted in dietary restriction even when there is a global reduction of nutrients in the periphery. But not only, because interestingly, these mice under dietary restriction, as you can see here, from day 0 of the diet protocol, and here from the beginning of the infection in this bar, they lose less body weight compared to the ad libitum control groups. They only lose around 5% of the initial body weight compared to 18% of the ad libitum mice. And this is followed by less glucose consumption overall. So if you compare the glucose these mice they are using from the beginning, the serum blood glucose, you can see the dietary restricted mice, they are dropping only around 15% of the initial blood glucose, while the ad libitum mice, they are dropping around 45%. This is true for other types of infection. So we infect these mice with Citrobacter rodention following the same dietary protocols. And after 10 days, we look for the same parameters. We can see that they survive the same. They have the same bacterial burden in the feces and the spleens and also other lymphoid organs. And there's a preservation of the body weight and also in the blood glucose consumption on these mice, showing that the immune response is upheld in the context of a dietary restriction while using few nutrients. But how is this balance maintained, right? So once these infections, they require strong CD8 cell response, as I said before, we assessed how the cells they were doing a dietary restriction, and for that, we infected mice with Yersinia, and after 8 days, which is the peak of the infection, we accessed the specific Yersinia cells, especially for the YOP antigen, using a tetramer staining. So as you can see, the dark green dots, they represent the adult group, the control group, and the light green dots, they represent the data restricted group. And you can see they have way less CD8 cells activated against this bacteria at the peak of the infection, as you can see as well in these dot plots. And we also observed that these effector cells isolated from the dietary restricted mice, they have a reduced potential to proliferate, as you can see in the Ki-67 staining. However, they are producing the same levels of cytokines as interferon gamma and TNF-alpha, even in percentage or MFI. And this is true for other factors as IL-2 or granzyme B as well, which is important for cytotoxic CD8 cells. This shows that the effector T cell proliferation is impaired during a dietary restriction. However, there is no impact in their function, which is quite interesting. And to check which specific genes could be modulated in the dietary restriction, we just sorted these tetramer-specific cells by flow cytometry, and then we did an RNA-seq for the cells. So something that got our attention was the number of genes and pathways related to the response to steroid hormones. And at this point, we had a question because could this be acting in CD8 cells as well, or was just the reduced amount of nutrients which were impacting the CD8 cell proliferation? And we know already that a class of hormones, steroid hormones that were increased in the dietary restriction were the glucocorticoids because of Nick's 2019 paper. And as you can see, even in the serum and also at tisular level, the dietary restricted mice, they have a way higher production of corticosterone. So, well, glucocorticoids, they are quite interesting molecules, at least in my point of view, because they can exert a lot of functions. And they are produced by the cortex of the adrenal glands in a circadian basis, especially at the morning. And they can act a lot of, they can make a lot of actions in some contexts, as for example, in fasting and exercise, where they increase their production to tell the muscles and the liver to use less glucose, which is important for maintaining the glucose homeostasis during this fasting or exercise. And for the immune system, needless to say, there's a lot of studies trying to check the effects of glucocorticoids on it, right? So what I can say to you guys now is that low doses of glucocorticoids, they can benefit memory generation. So it's good for CD8 and CD4 cells to generate memory and also to express bone marrow homing markers. On the other hand, if you have an excess of these molecules, they can be very immunosuppressive. That's why you see immunosuppression caused by long-term stress and also when a person is using synthetic glucocorticoids in clinics. So, well, let's see if these effects on the CD8 cells, they are caused mainly by the reduction of nutrients or because of the glucocorticoids. That's why we did this in vitro platform. We isolated these YOP1 transgenic CD8 cells, which are coming from a YOP1 mouse. It's quite similar to OT1 mice. They only have CD8 cells against a specific antigen. In this case, it's against the YOP antigen from Yersinia. And then we activate the cells in vitro with the same amount of nutrients in the presence of this YOP peptide, but we put, for some conditions, serum from mice fed ad libitum or control mice, serum from mice under dietary restriction or eating less, or with dexamethasone, which is a synthetic glucocorticoid. And after 5 days of activation in the presence of IL-2, we look for the proliferation of the cells. So as you can see, even having a similar access to nutrients from the beginning at the culture, the CD8 cells that were put in contact with the serum of dietary restricted mice or in the presence of dexamethasone, they proliferate way less, shown by the expression of Ki-67 here in comparison with the control mice treated with the ad libitum serum. And it's also reflected in the number of cells we have in the end of the culture, showing that glucocorticoids present in the serum of the dietary restricted mice could be the factor behind the CD8 dampened proliferation. Similarly, if we treat some ad-libitum-fed mice, so they ate whatever they wanted, doing all the protocol, with a very low dose of dexamethasone for 2 weeks, and then we infect them with Yersinia, following the same protocols for dietary restriction, they also have way less tetramer-specific CD8 cells, and the blood glucose variation is also lower for them, showing that they can preserve the blood glucose, if they are treated with glucocorticoids. Okay, so at this point, we wanted to know what is the physiological effect of dampened CD8 cells, even in a context where you need them, right? Especially for Yersinia infection, Citrobacter infection, they need the CD8 to fight the bacteria. So here we did an intervention as follows. We took some black 6 mice, we put them onto the ad libitum or dietary restriction. We infect them with Yersinia, and from day 0-1, we took some EOP transgenic cells from other mice, and we knocked out 2 different genes and 2 different groups of cells. The CD19, that for CD8 cells is not a very important gene and also codifies the protein with the same name, or the NR3C1 gene that encodes the protein with the same name, and it's the glucocorticoid receptor. So at day 5, which is the time point we found where the CD8 specific to cells, they start to pop up in these mice, we inject the cells back to the system. And this is how the blood glucose they look like. So you can see for the ad libitum mice, once they have increased access to food, they don't have any difference. However, mice under dietary restriction and the light green colors receiving the CD19 knockout cells, they can control better the glucose rather than the others receiving the glucocorticoid knockout cells, showing that responding to glucocorticoids during a dietary restriction restrain the CD8 cell expansion and also how the mouse can use glucose in this context. Okay, but now we have a conceptual problem, right? Because after the primary challenge and after the contraction of the immune system, you have to generate memory. But what about memory generation? If you have less effector clones, it's memory maintained in the dietary restricted mice. So just a brief comment on how CD8 cells, they can generate memory. So when a CD8 cell becomes a factor, it can have 2 fates. One possibility is for them to become a memory cell. So they start to express in high levels of this marker, CD127 or IL-7 receptor. And then we call the cells MPECs or memory precursors effector cells. Or they have another fate, which is expressing high levels of KLRG1. And then we call the cells short-lived effector cells that are gonna die after the effector response, and they make part on that contraction I have shown before in the beginning of the presentation. So let's look like how these populations, they look in the ad libitum and the dietary restricted mice. So we did the same protocol. And here are the graphs showing the percentage among that effector cells, which are MPECs or memory precursors, or SLAGs or short-lived cells. You can see the dietary restricted mice even have less effector cells. These effector cells, they are more prone to become memory precursors, as seen in these blue gates showing a higher expression of the IL-7 receptor. And just to check if the glucocorticoids were making this effect as well, it just treated that mice with a very low dose of dexamethasone for 2 weeks. We infect them, and then you can see we have phenocopy. So now mice treated with dexamethasone during infection, they have more memory precursors and less short-lived cells. And just to see if this was really true, when we knocked out that glucocorticoid receptor and gave them back to the dietary restricted mice, you can see we ablate this phenotype. So cells which are still expressing the glucocorticoid receptors, they can become MPECs easily. However, if they don't have the glucocorticoid receptors, they are not expressing the IL-7 receptor, showing that for the memory generation, it's important for the dietary restricted mice to have increased levels of glucocorticoids and thus increase the expression of IL-7 receptor in the CD8 cells. So here's just showing the same scheme in vitro by isolating that EOP transgenic cells, and then I activate them in the presence of ad libitum and dietary restricted serum or with dexamethasone, and you can see that there is a significant difference between data restriction and for dexamethasone is way higher. So it shows that glucocorticoids can make this effect on CDH cell. So what I have shown so far is the reduction of calories during a dietary restriction and thus the change in the eating behaviors that culminates with reduced levels of glucose in the body. They increase the production of corticosteroids in the dietary restricted mice, and they can exert direct effects on the CDH cell expansion. While in the ad libitum mice, the CD8 cells, they have increased access to nutrients, and thus they can proliferate a lot. On the other hand, the dietary restricted mice, they have a reduction on the expansion and the total numbers of the effector T cells, which isn't necessarily related to a lack of function because they are producing the same levels of cytokines and other factors. Another interesting point is that despite the effector reduction, they can still enrich for memory precursors, which maintain the memory response over time. And however, at this point, something puzzled us because I've shown at the beginning that diet-restricted mice, they have a maintained protection against the bacteria, right? So considering that the CD8 cells, they are very important to fighting Yersinia infection and Citrobacter infection, how is the protection upheld during a dietary restriction? So at this point, we thought that this fact suggested a compensation by other immune cells. So we went to check. And in fact, the only population that was enriched in the blood and in the periphery of these mice during the dietary restriction were neutrophils. So as you can see here in these graphs, they correspond to around 50% of cells in the blood of infected dietary restricted mice. And the same is observed for other type of infections as for Citrobacter, as they correspond to 30%, which is like 3 times more compared with the ad libitum mice, showing that neutrophils are increasing during a dietary restriction against this type of pathogens. And not only numbers, but when we did an RNA-seq from these cells isolated from mice ad libitum or during dietary restriction, we saw a lot of pathways upregulated to a positive control of the immune response. So you can see a strong signature for inflammatory cytokines and killing by these neutrophils. So to assess if this neutrophil function were changed during a dietary restriction, we did this in vitro experiment where we isolated neutrophils with magnetic beads from the ad libitum mice or dietary restricted mice, and then we put them in contact with Yersinia for 1 hour to the cells to phagocyte the bacteria. At this point, I did flow cytometry, and we saw that the amount of bacteria phagocyted by this time was the same between ad libitum and dietary restricted mice. And then we put some gentamicin in the media to kill the extracellular bacteria. So after 2 hours, we lysed the remaining cells, and this was the result for the killing assay. So as you can see, neutrophils isolated from the dietary restricted mice, they have way less bacteria when compared to the ad libitum mice, which show that even uptaking the same amount of bacteria, they are killing better this bacterial content. Okay, so let's do a functional assay to see how the absence or reducing neutrophils in vivo can affect these mice during a dietary restriction. So we did the infection protocol, and after 5 days, we gave them 2 shots of anti-Lys6G at day 5 and day 6, and then we look for the survival of these mice. So here you can see for the ad libitum mice that are in purple here, they don't have much difference because from day 5 on, as I said before, they are producing a lot of CD8 effector cells, producing cytokines. But for the dietary restricted mice in lilac, they start to die. So neutrophils are even important for dietary restricted mice, even in the time point where these mice, they would be generating CD8 cell. Okay, but I've shown before that glucocorticoids, they have a strong impact on the immune system, especially on the CD8 cells, right? But can glucocorticoids make some effects on neutrophils as well? And when we were trying to understand the effects of glucocorticoids on CD8 cells, I found some works that were showing the effects of glucocorticoids on neutrophils, but positive effects, as in this one from the 70s showing that patients treated with dexamethasone, they present neutrophilia over time. And this one from the 90s showing that when you take some human neutrophils and treat them in vitro with several classes of glucocorticoids, they increase their lifespan. so they apoptose less. So apparently, glucocorticoids, they have a positive effect on neutrophils rather than negative as for the CD8 cells. Okay, so let's check if the glucocorticoids are making this on the dietary-restricted serum. For that, I took the whole CD4/5 positive cell content, and I put the cells in the culture for 2 days with the serum of ad libitum or dietary-restricted mice or with dexamethasone. And this is how they look like. If you look for, after 2 days in the culture, only 2% of the cells remaining in the culture for the ad libitum treated cells, they were neutrophils, while the dietary-restricted mice treated cells, they had around 5 times more neutrophils. And for dexamethasone is way expressive. It's like 90% of the remaining cells, they were neutrophils. And here, I must emphasize that dexamethasone is a synthetic glucocorticoid, so it binds to the receptor with an affinity that is 30 times bigger than the endogenous corticosteroids. That's why we see a strong phenotype like this. And indeed, if we treat these mice ad libitum fed, so they ate whatever they wanted, they had the same access to nutrients, but then we gave dex to them, and then we infect these mice, you can see they also have more neutrophils in steady state or during the infection, and they also kill better bacteria when we isolate these neutrophils from these mice and submit them to a killing assay with Yersinia. Okay, so let's consider that glucocorticoids, they are important to maintain these neutrophils alive, and that's why the dietary-restricted mice, they have a lot of neutrophils, even though they don't have the same level of nutrients. So what happens if we disrupt the glucocorticoid production in these mice? And that's why we use this drug, which is called metirapone. Metirapone, or GCI, it's a glucocorticoid synthesis inhibitor. So why it's very difficult to deplete totally glucocorticoids. We start this treatment 1 week before the infection of these mice, and then we kept this metirapone in drinking water for the whole experiment. And it's interesting because if I look at day 5, how the cells they are looking like in the blood, you can see the expression of active caspase 3 in neutrophils from the blood of dietary-restricted mice treated with the glucocorticoid inhibitor. They have a higher expression of caspase 3, indicating that the cells, they could be dying at this point. And this is true because if you look for the dot plots here from mice treated with the GCI, even for the ad libitum, it happens as well. They have a 2 times reduction of the neutrophils in the periphery, but for the dietary-restricted mice, it's even more significant. They have 5 times less neutrophils when compared with the counterparts controls. And this all culminates with a reduced survival for the dietary-restricted mice, similarly to what I have shown before to the anti-Lys6G treatment. So as you can see, ad libitum mice, they are not affected, but for dietary restriction, they start to die here. So it seems that glucocorticoids, they can promote this neutrophil survival during a dietary restriction, and that these neutrophils, they are critical for the host protection. But the question I had at this time was, neutrophils, they are highly energy-dependent cells, and they use a lot of glucose to maintain themselves. So why you are enriching a population that uses a lot of glucose in the context of reduced glucose and other nutrients availability, right? It doesn't make a lot of sense. So far, we know that neutrophil metabolism, it relies mainly on glycolysis because they don't have a lot of mitochondria compared to other immune cells. They just have a few mitochondria in the cytoplasm, so they are not very good at OXPHOS. And this means that low glucose availability could affect how the cells, they are activated. But something that was impressive is that when we look for that RNA-seq I've shown to you guys before, some of the downregulated genes and pathways were related to the glucose homeostasis. And this reflected as well in how the cells were uptaken to NBDG, which is a glucose analog. So it means that neutrophils from mice during a dietary restriction, they are uptaking less glucose or less glucose analog in this context. And this is also seen here in this extracellular acidification assay, where you can see the ad libitum neutrophils, they are highly glycolytic, but when you perform the same assay with the dietary-restricted neutrophils, they use way less glucose from the beginning, and the glycolysis is lower for these neutrophils at the end of the assay. Okay, so let's check if the glucocorticoids, they can be doing these activities on the neutrophils, right? And that's why I isolated these neutrophils with histopachic gradient, and then I put these neutrophils in vitro in the presence of adlepton or data-restricted mice serum, or with dexamethasone. And you can see by the 2-NBDG uptake that even the neutrophils treated with the data-restricted mice or treated with dexamethasone, they uptake less glucose analog, and also they consume way less glucose in the media at the end of the experiment. It's visible. I can see in the plate how the cells, they are using less glucose at the end. So altogether, this data suggests that glucocorticoids, they can, beyond promoting the activity of these neutrophils and their survival in the periphery, they can also maintain the energy balance, reducing the uptake of glucose, and somehow making them use another nutritional source, right? At this point, we thought that the mitochondria could be affected as well because it was the first bet we had at the time. But we start to look at the OCR assays, the mitochondrial potential and mass, the lipid uptake, and it was all the same. So it didn't have any difference. So at that point, we had this autophagy reporter mice in the lab, and we put them onto adlepton or dietary restriction protocols for 3 weeks. And we can identify the autophagy, the cells undergoing autophagy as follows. The total content of LC3B, which is a protein, which is very important for the autophagic process. It's EGFP positive, but when you start to go into the autophagosome and the autophagolysosome, it starts to quench the GFP protein and start to express the RFP protein. So that is what we call early and late autophagy, which is more important because it indicates for me the cells that were undergoing the autophagic process at the moment of the euthanasia. So as you can see in this dot plots and the graph, mice treated on the protocol of data restriction, they have 3 times more cells undergoing autophagic processes compared to the adlepton mice, which shows that somehow it seems that they are taking less glucose, but they go better under the autophagic process, and they may be using their own organelles to produce and use energy in this context of reducing the nutrients in the periphery. So to sum up, in the reduction of calories that happens during a dietary restriction and thus the reduction of glucose and other nutrients availability, there is increased production of corticosteroids that culminates in a rewiring of the innate and adaptive immune system during the primary infection. So for T cells, it's characterized by the reduction of the proliferation and expansion of the cells in the effector phase, but it isn't necessarily related to a lack of function because they are producing the same amount of cytokines and other factors. And furthermore, these glucocorticoids, they can also improve the generation of memory by the CD8 cells. At the same time, on the neutrophils, the glucocorticoids, they have an opposite effect because they increase their lifespan and also the circulation and the dietary restriction mice, and then they fight better the infection in the scenario of reduced CD8 cells. So the nutrient consumption of the cells is also rewired because they start to use less glucose, which is the mainly nutrient source, and they start to do a lot of autophagy, which maintain the cells alive for a longer time. And altogether, we think that this is an evolutionary mechanism that happens during a dietary restriction or reduced access to food and nutrients. And we propose that this is critical for helping the host to fight infections while also avoiding malnourishment, which means upholding the immunity with an energy efficient way. So they can control the infection the same, but using less energy. So that's what I had to show you guys today. So I want to thank all the people behind this work, especially the New York group, my advisor, Nick Collins and Sanji Han, and my lab partners, Mingam Jong and Charlie Scar, my advisor here in Brazil, Nils Olsen, and also FAPESP for giving the fellowship for this abroad student, and all the people that collaborated within the Jill Roberts Institute and Friedman Center for Nutrition and Inflammation. And thank you so much for watching the presentation as well.
All right, thank you, Luísa. So please, we open for questions for the students first. So we have a question here. Anybody in Rio that should have a question too? So let's start with Bernard.
Thank you, Luísa. Beautiful presentation. I was wondering, I have two questions actually. First one is you show that dietary restriction lessens the effector cell and increase the memory CD8 T cells, right? Is there any difference between adiliptin and dietary restriction in the phenotype of these cells? Are they better or worse? And the second question is kind of like that. If you try, what happens if you re-challenge them with your senior, like a chronic, you know?
Yeah, yes, I did. I just didn't show in the presentation, but phenotypically, they don't have a lot of differences. If I look on the interferon production and TNF production, they are producing the same levels. And if I do a late challenge, even vaccinating these mice or infecting a first time and then a second challenge, they gonna expand the same. So I look like for the memory response and the secondary challenge, they have the same levels of CD8. The thing is different only for the primary challenge. So phenotypically, they are very similar, but in the memory challenge and in reinfection at a longer time, they have the same response.
Right. So Bruno has raised his hand there. Is there a question from students there, Bruno?
Well, actually, not from students, but Miriam would like to follow up on this question to Luísa.
Okay.
Okay. Diego, is that okay, or do you have other students?
Oh no, it's fine.
Yeah.
All the other questions here, at least so far, from PI. So.
Okay, thank you, Luísa, for the talk. So I'm gonna follow up. I think it's Bernardo, right? The name of the student. I was gonna ask a similar question, but related to repertoire, because we know that you can skew the T cell differentiation if you use less TCR engagement. So for the same clone, for instance, OT1, right? If you use less anti-CD3, you can get memory cells. If you have more anti-CD3, you get effector cells. And it looks like the enhanced glucocorticoids might be dampening the response, and you might be then getting more memory cells, but with the repertoire that would be effector cells, and that would explain why your second response is similar in both mice. So I wonder if you looked at that and if that could be the reason why you have more memory with less effector cells.
Thank you for the question, Miriam. So I didn't look at this at my project, but another student at the Nicolin's lab, she was looking in, especially about this and TCR engagement and the downstream pathways during a dietary restriction. So I still don't know which results she has, but she was looking at this. I think it makes a lot of sense.
All right, thank you. Thank you very much.
Thank you.
Luísa. Congratulations on your work and also for the active student you are in our program. So I'd like to register that here. So I have a few comments that in my concept is a dietary restriction leads to immunosuppression in a long term or
it depends on the population because, for
Yeah.
example, it has some papers showing that for monocytes, even though they have decreased numbers of monocytes, they can act better. So it suppressed immune response at certain point. For lymphocytes is a strong suppression because for B cells, CD4 cells, and CD8 cells, we see the same response, but for myeloid cells is way trickier. Because for dendritic cells as well, they have the same levels of them in the gut and the lymph nodes, but they are acting better. So it depends on the population.
Yeah, my point is more like, just to keep on the topic, there's no
Yeah.
free lunch, right? Like
Yeah.
you're reducing your intake,
Yeah,
and at some point it's going to be negative for your body. Like
yeah,
otherwise, people
Exactly.
starving would be as immune competent as the
Yeah.
non.
for other
So-
systems as well, right? Not only for the immune system, but
Exactly.
I-
So, and my point is like, have you tried, I don't know your bacteria, like if there any strain that is like more that stronger disease causes stronger disease, like more pathogenic.
Yeah.
Do you think that like you would see the same phenotype or increasing the amount of bacteria? I don't know. I'm gonna say like playing the amount and or this type of strain. If you would see the same phenotype, like.
Yeah, I've tried to give higher doses of Yersinia at the beginning, but it was bad even for Adlepton and DR mice. We couldn't get to a dose that like it's limit between it. So I think I didn't test other stronger infections other than Yersinia, but it's a question I also have regarding other types of infection, especially for viral infections that also requires a strong CD8 cell response. I don't know if neutrophils would be good in this case because they cannot phagocyte the particles, right? So I think it's a very good question, but I haven't accessed a stronger bacteria at this point. Yeah. Thank you, Anderson.
Right, so I have a question from Leandro here, then Bruno and Elizabeth. Actually, Elizabeth is a student, right? So let's start with her.
Hello, can you all hear me? Or
Yes.
Yes.
yes, okay. Thank you so much for the talk. Really amazing work. I am in the microbiology program actually, and I would like to understand a little bit more of why is that in the dietary restriction model, the bacteria seems to be more unable to replicate inside the neutrophils. I didn't quite get that. Could you discuss that a little bit more? Thank you.
Yeah, so something I didn't have time to show is that a lot of pathways related to antimicrobial peptides, they are also increased in this neutrophil. So somehow they can kill better these bacteria within the phagosome. So we think it could be this. We cannot prove this so far, but we have only seen these antimicrobial peptides upregulated in the RNA-seq, but we think it can be this mechanism. Because we've checked the production of HOS, for example, and it's the same. So it doesn't justify why these neutrophils, they are killing better bacteria. So I think it can be something related to the antimicrobial peptides. I can answer this question for you so far. Thank you.
Right.
Thank you.
Bruno, do you have a question from a student there or? No? So, because Clarissa also raised her hand online.
Clarissa is not a student.
So,
Okay.
no,
So
but so I can
I'll
wait
respect
a
the
bit.
argument, and then I'll give the mic to Leandro, and then you guys, and then Gustavo, and then Clarissa. Okay.
Congratulations, Luisa. It's very nice work and very interesting. So I have at least two questions. So the first, because I work with neuroimmunology, how about the norepinephrine, right? Did you guys check this? So because norepinephrine is very important for immune response to immunosuppressive. And another question is about the viral infection, right? Because there is like a paper from Rudensk lab. Probably you know this paper, right? That they show there is different outcomes from mice that are viral infected or bacterial infected with different.
So for the first question, no, I didn't check the norepinephrine. And the second question is, I think it would be completely different because, as I said to Anderson, I think neutrophils, they are not efficient to viral infections. So that's why the Rudensk paper, they have that outcome, and this paper we have this outcome because mostly we used bacteria. We also tested listeria for an infection. We have similar outcome, but we didn't use a viral strain. So I think it would be completely different, even for fungal infections and other types as well.
Just another one, just to complete. Did you guys check the TCF1?
Yes.
In these CD8 T cells?
That are restriction mice.
Okay.
Yeah. It looks like when they are affected, they decrease the expression, but the GR, they go into the memory phenotype very fastly, so they start to express again TCF1. Yeah.
Right. So, Bruno, you want to go ahead?
Sure. Thank you, Diego. Thank you, Luisa. very nice talk. Very interesting. It's really good to see that glucocorticoids are still a source of new data and very exciting data as it is. So I was wondering, one, if you continue your dietary restriction for longer, and what it happens. And two, if food restriction is a cyclic phenomenon, right? So even if it's just for the seasons, but even for mice, small animals, they don't have food security, right? So it changed a lot throughout. So have you ever tried to do the restriction, return to ad libitum, and then do the restriction again and see what happens, if there are any lasting effects?
That's exactly what I'm doing right now. So I intend to put this as part of my postdoctoral fellowship as well, basically trying to understand what happens after. So basically the longer time point we looked for this mice with foreign fault infection was after 6 weeks of this dietary protocols. But I've performed some experiments, putting them back in the control diet and then checking how the cells they are. And it's incredible because especially for the CD8 cells, they start to express a lot of apoptosis receptors. And the memory cells of the bone marrow, they start to go. So it's crazy. Like, to be in a dietary restriction for a time, it can shape how your metabolism and your body is for a longer time. So this is something I want to keep studying. I still don't know the answers because I didn't perform these experiments for this paper, but it's something I want to keep working on it.
Right. So it's very exciting because what is normal, right? So it's to have food all the time, or to have all these variation. I think throughout evolution we were exposed to a lot more variation than what we have now. Right.
Yeah.
Thank you.
Thank
Congratulations.
you so much, Bruno. Thank you.
Hi, Luísa. Excellent work. Not unexpected, right? I have a few comments. One is following Bruno's. So it looks that dietary restriction to our society is something like bad, but I don't think it's the same thing as malnutrition.
Yeah.
Right? So, and if we look at the evolution, then dietary restriction is how we survive.
Yeah.
Okay. So actually, I understand from your work that the normal way we are eating is not good. So it's detrimental. It's not.
Yeah.
You see?
I think not necessarily, because like first point is that the protocol we used for these mice is extreme. So they were eating like in the borderline of being malnourished. So 50% less. Usually protocols they use for dietary restriction is 20 to 30%. So I think it's different because in the context of like a abrupt reduction of the nutrients they have to very low levels. So this is what I'm trying to show here. I don't say that dietary restriction it's bad, because like there's a lot of literature showing the opposite, but I feel like it depends on how you are doing that. If you are doing dietary restriction all the time, and it's what we call the yo-yo effect, right? You go back to the normal diet and then you go back to the diet. And this is something that also interests me a lot. It can be bad for you. So you have to maintain this food protocol for a longer time for having its beneficial effects, rather than keeping back to the normal diet. It's something like that.
Right. No,
Yeah.
I agree, but I think the point that is important, and I was getting maybe in a different way, is whether this dietary restriction in the long term for the mice is bad physiologically, or this is the borderline that is still positive. Because if it's malnutrition, then it's a different thing.
Yeah, totally.
So you're saying that, so your answer for that?
This type of protocols, like 50%, they maintain the body fat and the body weight in a plateau at the end of the experiment. So they are not becoming malnourished.
All right.
But yeah,
okay. So
in lower dietary restriction protocols, it can be.
Okay, so that's good.
Yeah.
So another thing that I want to ask you is regarding to thymocytes. Okay, so have you looked at the,
Yes.
production of naive cells? So you measure proliferation, right? And compare effect to memory. But what about the naive compartment?
Yeah, that's a great question because, for example, I've read in the literature before that a high dose of dexamethasone can also kill thymocytes, right? So it was a question for me if they have the same variability of TCRs within these mice, because if these naive cells, they are dying, how they can fight the antigen in the future. So we track the TCR on these mice. When we saw that the naive T cells, they were going to the bone marrow, but they actually have the same diversity of TCRs, which doesn't seem a problem. So they can find, like, in terms of number, the same amount of antigens when they go out of the thymus. I think like this level of glucocorticoids and this time point we were looking, they were not sufficient to kill thymocytes or to reduce the diversity of TCRs.
Okay, that's very important. And the last thing is related maybe to it's two aspects of perhaps the same question. So many other stress induces glucocorticoid.
Yes.
So all stress would lead to protection. I don't think so.
No.
And then also I noticed from your data that if you get the ad libitum mice and inhibit the glucocorticoid signaling, there's no really any effect.
No.
Only if you get dietary restriction,
Yes.
which suggests that you have a second signal, second event, any other thing that is detrimental, but the glucocorticoid is holding that back.
Yeah.
So what do you?
So this is an interesting point because when we were treating these mice with the glucocorticoid synthesis inhibitor, we are given a higher dose from the beginning. But then the ad libitum mice, they were not surviving as well. And then at in vitro experiments, we found that some levels of glucocorticoids are necessary to activate the CD8 cells from the beginning as well. We don't understand why. If you completely deplete the glucocorticoids in the in vitro activate the CD8 cells, they don't become activated. So it's something we still don't understand. I don't know why, but I really feel like for the ad libitum mice, glucocorticoids, they could be important as well, not only for dietary restriction. And I feel like for the first question regarding like other types of stress that could cause an increase in glucocorticoids, I think it's a different context because in here you have increased glucocorticoids, but you have enhanced access to glucose and other nutrients. So if you have increase in glucocorticoids and you have low levels of nutrients, this is the response. But if you have increased access to nutrients, somehow you can see something similar what we see in the Cushing syndrome or in other contexts where you have like increased glucocorticoids and then you become insulin intolerant and you have like reduced tolerance to glucose. I think it like affects all these scenarios because you are in a context where you have enhanced access to food, but then you have increased glucocorticoids.
Yeah. So the two combination is very bad for us.
Yeah.
So a lot of stress, a lot of food.
Exactly. Yeah. Yeah.
All right. So Clarissa had raised her hand before. Do you want to ask, Clarissa?
Yeah. Yeah.
Okay.
So thank you, Luisa. It's a very, very good work. And I'm going more like Bruno for the metabolic side. Have you thought, because you are doing dietary restriction, just cut half of the diet. Have you thought of playing with the diet, like cutting half of the calories with taking out half of the carbohydrates or lipids to play with this? So if you have the glucocorticoids response plus high glucose, would this be different, like the same as the ad libitum? Or like if you give more lipids or less, if you cut on the lipid side or on the protein side, I think this could change this scenery.
Yeah, I agree with you. That's a very good suggestion. I haven't tried like playing with the nutrients. The only thing I've made was trying different percentages of reduction of the food. So like from 20% to 50%. And we see that it's proportional the amount of corticosteroids these mice produce, as well as the amount of neutrophils they remain in the bloodstream and also they preserve. So I think like if I play with the nutrient, it's gonna be a very good way of assessing how is the importance of glucose to this phenotype, right? Because I'm very going towards the glucose, but I think other nutrients can impact as well once they have like lower levels of amino acids and proteins and lipids as well. But we couldn't prove this so far for the neutrophils.
And yeah, also interesting is to look at the food preference, because there's a good work from Suzanne Lafleur in the Netherlands, where she was working. This works better with rats when you do ad libitum, but then you separate and then you check if the mice go more for the sucrose or more for the lipids. You put separated the diet. You separate the carbohydrates from the lipids. And then you check the preference.
Yeah.
So would it be that the infected mice would prefer to go to sugar or to lipids? Would they try to compensate the sugar diminishing after the diet? I don't know. Many things that are coming as ideas.
Yeah, but
You
I
can
understand your
spend
question. I think it
your whole
totally
life.
makes sense.
Yeah.
I feel like
would.
I don't have these results, but if I would say to what I think about it, is that they would be more prone to eat glucose or something related, because the bacteria also wants that. So it's something be related to sickness behavior, right, in this mice. But I cannot say that.
And then, and then maybe this would counteract the good effect of the corticosteroid that you're saying.
Totally. Yes,
So if
exactly.
we are sick and we go for the sugar, because you have a craving sugar that some virus gift, maybe this is the worst.
Yeah, exactly.
Yeah, just play with it. It's very, very interesting. Thank you.
Thank you so much for the suggestion, Clarissa.
Right. Thank you, Clarissa. Bruno also raising his hand there.
Sure. I just have a comment that may or may not be useful, but you know that glucocorticoids, they do not signal only through the glucocorticoid receptor. They also are very the strong ligands for the mineralocorticoid receptor. So for some of the functions of glucocorticoids, particularly the feedback regulation, mineralocorticoid can be a more important pathway of signaling. I don't know if in the immune system mineralocorticoid receptors are present, but it's something that you may check, because the response may vary with the actual concentration of glucocorticoid that you can achieve in the circulation, and then the response may go through more glucocorticoid, or it also includes the mineralocorticoid response.
Yeah, I don't know about the expression of mineralocorticoids receptors in the immune cell, so it's something I have to check, but it's a great suggestion.
All right, thank you. So I guess I have no further questions. Thank you again, Luísa. Congratulations for the work. You can see everybody